Hello everyone, welcome back to another video on Chapter 3, the movement of substances across the plasma membrane. We will be studying the effects of hypotonic, hypertonic, and isotonic solutions on animal cells and plant cells. Here is an overview of what we will be studying. When an animal cell is placed in a hypotonic solution, which is more dilute than its cytoplasmic fluid, it will undergo lysis or bursting of the cell because there's net movement of water into the cell by osmosis, and the cell will swell up and burst. For red blood cells, this is called hemolysis. When an animal cell is placed in an isotonic solution that has the same concentration as its cytoplasmic fluid, water will diffuse in and out of the cell at the same rate. The cell will maintain its normal size. When an animal cell is placed in a hypertonic solution that is more concentrated than its cytoplasmic fluid, there's net movement of water out of the cells by osmosis, and the cell will undergo crenation, which means it shrinks as it loses water. For plant cells in hypotonic solutions, water will diffuse in by osmosis, and there's net movement of water into the cell by osmosis, and the cell will become turgid. The plant cell will maintain its basic shape because of the rigid cell wall. When a plant cell is placed in an isotonic solution that's the same concentration as the cell sap of the plant, water will diffuse in and out of the cell at the same rate. So there's no net movement of water through the plasma membrane. No net movement of water into or out of the cell, and the cell is flaccid. When a plant cell is placed in a hypertonic solution, which is more concentrated than its cell sap in the vacuole, water will diffuse out by osmosis. There's net movement of water going out by osmosis, and the plant cell will undergo plasmolysis, meaning the cytoplasm will shrink and the plasma membrane will pull away from the cell wall. Let's revise the osmosis rule once again. It says that water always diffuses from a hypotonic or dilute solution to a hypertonic or more concentrated solution, right? So if we have two solutions, A and B, A has higher concentration of water molecules here, or higher water potential, but lower concentration of solute. B has lower concentration of water, but higher concentration of solute. So we say that B is a hypertonic solution, more concentrated with the solutes. A is more dilute. It has less solutes, but more water, right? So A is a dilute solution or hypotonic solution. B is a concentrated solution or hypertonic solution. So water will always diffuse from the hypotonic solution to the hypertonic solution, like this. The water molecules will not remain as they are, but they will always move from hypotonic or diffuse from hypotonic to hypertonic solution. So in the end, B will gain water and A will lose water. Let's discuss the three types of solutions which have different concentrations when compared to another solution. The first one is the hypotonic solution. The word hypo means under or less than. So a hypotonic solution is a solution with a lower solute concentration compared to another solution. Take for example these two solutions here. A has one sucrose molecule, the green molecule here, and B has 3 sucrose molecules. So we say that A is hypotonic to B. Lower solute concentration, lower number of dissolved particles, okay, for the same volume of water. However, a hypotonic solution having a lower solute concentration means that it has higher water potential. So the water potential and the solute concentration are the opposite of each other. If you have a solution with lower solute concentration, you will have higher water potential. For example, you can see here 10 water molecules, and this side there are 5 water molecules. Okay? So, in other words, in layman terms, we consider this hypotonic solution a more dilute solution when compared to another solution. Let us take the example of 2 cups of coffee. Okay? The first cup of coffee is made by dissolving 1 teaspoon of coffee in a cup of water. But the second one, you use 3 teaspoons of coffee dissolved in 1 cup of water. So the one with 1 teaspoon of coffee is the more dilute solution compared to the one with 3 teaspoons of coffee. So the one with 1 teaspoon of coffee is the hypotonic solution compared to the other one with 3 teaspoons of coffee. Right. So example is distilled water is hypotonic compared to 5% sucrose. So distilled water is 100% water molecules. It has no dissolved substances. Thus, it's a very dilute solution and is the most dilute that you can get. So it's hypotonic to a solution that has 5% sucrose in it, meaning 5 grams of sucrose per 100 ml of water. Right? And distilled water is hypotonic to the cell sap of plants because cell sap of plants contain some glucose, mineral salts, and amino acids. So distilled water is hypotonic or more dilute than it. Let's discuss the second type of solution, which is the isotonic solution. The word iso means the same. An isotonic solution is a solution with the same solute concentration and the same water potential compared to another solution. Referring to these 2 diagrams, solution A and B, solution A has 2 molecules of sucrose and solution B has 2 molecules of sucrose also. Also, the number of water molecules are the same, 7 here and 7 there, right? So, as an example, 5% sucrose solution or 0.5 m sucrose solution is isotonic compared to the cell sap of plants. So, from experiments, it has been found that this 5% sucrose solution, or in molar is called 0.5 molar sucrose solution, has the same concentration as the solutes in the cell sap in the vacuole of plants. Okay, so we have to remember these 2 values. These are the isotonic solutions to the cell sap of plants. Lastly, we'll discuss the hypertonic solution. Hyper means above or more than. So a hypertonic solution is a solution with a higher solute concentration than another solution. Again, referring to diagram A and B, diagram A has 3 molecules of sucrose compared to B, which has only got 1. So solution A is hypertonic compared to B because it has higher solute concentration. But in terms of the water potential, it has lower water potential. Okay, so hypertonic solution has lower water potential or lower concentration of water compared to another one. Okay, here you have 5 molecules of water and here you have more molecules of water. In other words, a hypertonic solution is a more concentrated solution than another, more concentrated. Just like you make coffee, if you make a cup of coffee, you can make it dilute or concentrated. The concentrated one will have more teaspoons of coffee dissolved in the same volume of water. As an example, 10% sucrose solution is hypertonic compared to 5% sucrose solution. 10% sucrose solution has 10 grams of sucrose in 100 ml of water, whereas 5% sucrose has 5 grams of sucrose dissolved in 100 ml of water. So definitely 10% will be hypertonic compared to 5%, more concentrated. Now let's discuss what happens to red blood cells in the different types of solution. Firstly, red blood cell in hypotonic solution. For example, you can be asked a question like this. Explain what happens to a red blood cell, or maybe just a human cheek cell, in hypotonic solution or in distilled water, for example. What happens to the red blood cell when it is placed in distilled water? Right, how do we answer this question? Firstly, we must mention that the distilled water is hypotonic compared to the cytoplasmic fluid in the red blood cell. Distilled water is hypotonic compared to the cytoplasmic fluid in the red blood cell. Secondly, mention what happened to the water. What happens to water? Water diffuses into the cell by osmosis. Into the cell, osmosis. These are the keywords. Thirdly, explain what happens to the red blood cell. The red blood cell will swell up and then it will burst, because the plasma membrane is too delicate to withstand the osmotic pressure that builds up inside. These yellow arrows show the pressure from the water that is increasing in the red blood cell. Okay, so the plasma membrane is too delicate to withstand the pressure, osmotic pressure building up inside, and therefore it bursts and the cytoplasm comes out. So we put a lot of red blood cells in a test tube in distilled water. At the end of the experiment, after some minutes, you will see the whole solution will turn red in color because the cytoplasm comes out as the cell bursts. This condition or process is called hemolysis for red blood cells. Okay, hemo means blood, lysis means to break down or to destroy. But if it is just other cells like cheek cells, we just say lysis occurs. For red blood cell is hemolysis. So let us see how to answer this question about the hypotonic solutions. Explain what happens when a red blood cell is placed in distilled water. 8 marks. So you can start off by saying that the distilled water is hypotonic compared to cytoplasmic fluid of red blood cells. Don't just say distilled water is hypotonic to red blood cells. You must compare the two fluids, the distilled water and the cytoplasmic fluid, fluid to fluid. Okay, so distilled water is hypotonic compared to cytoplasmic fluid of red blood cells. Thus, water diffuses into the cell by osmosis, into and osmosis. All the words in red are the keywords. The cell swells up. Plasma membrane is too thin to withstand the osmotic pressure building up within. Thus, it bursts. And this process is called hemolysis, the bursting of red blood cells. And the cytoplasm of the cells is released into solution. Now, this answer is for essay question, 8 marks. However, in structured question, you do not have to write so long. You can take point number 1, hypotonic. Number 2, water diffuses into the cell by osmosis. 3, the cell swells up and bursts. And number 4, hemolysis. Okay, so it depends on the number of marks. Secondly, let's discuss the effect of isotonic solution on animal cells. When a red blood cell is put in an isotonic solution, such as 0.9% saline solution or 0.15 m saline solution, molar, water diffuses into the cell and out of the cell by osmosis at the same rate. Take note that the isotonic solution to the cytoplasmic fluid of red blood cell is 0.9% saline solution. There's the same concentration as the cytoplasmic fluid of red blood cell. Or if we are using the unit molar, it is 0.15 molar saline solution. So this unit is used in the textbook. Therefore, you have to remember these two values, 0.9% or 0.15 molar. They are the same concentration using different units. Okay. Normally, in layman's terms, we'll use 0.9% saline solution. Saline here means salt solution or sodium chloride solution. Okay, so When a red blood cell is put into an isotonic solution, such as these solutions, water will diffuse into the cell and out of the cell at the same rate. Therefore, the cell neither gains water nor loses water. No net movement of water into or out of the cell, and the red blood cell will maintain its normal shape and size. So isotonic solutions are used nowadays to prevent dehydration. If someone has lost a lot of water, you can take an isotonic drink which contains the same amount of salts and water as in the body, and he will recover, as his body cells will be able to function at a healthy and normal condition in this kind of situation. Okay, so the cell will maintain or retain its normal shape and size and be able to function normally. Thirdly, what is the effect of hypertonic solution on animal cells? So when a red blood cell is put into a hypertonic solution, such as 5% saline solution, which is more concentrated than the cytoplasmic fluid of red blood cell, water diffuses out of the cell by osmosis, right? From now, the red blood cell has a hypotonic solution. The cytoplasmic fluid is hypotonic compared to the solution outside, which has more dissolved substances like salt than it. So water will diffuse out of the cell by osmosis, and as the cell loses water, it will shrink or shrivel. And this process is called crenation, which is the shrinking of red blood cells. Okay, now you must be able to draw these diagrams also. For red blood cell in hypertonic solution, you have to draw the shape like, it's a bit like a star like that, with sharp points. And you must draw the size of the red blood cell smaller than the original one, as it loses water and shrinks. And also for hypotonic solution, you have to draw that the cell bursts. And in isotonic solution, you draw that the cell is the same size, retains its normal size. Okay, so here the hypertonic solution is 5% saline solution. How do we know that it's hypertonic? As long as the concentration of this salt solution or sodium chloride solution or saline solution is more than the isotonic solution. Just now we discussed isotonic solution for red blood cells or animal cells is about 0.9% saline solution. So 5% is definitely more than 0.9% saline solution, therefore it is hypertonic. There's more dissolved substances or more salt in it. Okay, let us go on to plant cells. Let's say you are asked this question: explain what happens when plant cells are placed in distilled water. Okay, a variation of this question is: explain what happens when the epidermal cells of onion are placed in, say, 1% sucrose solution. Okay, so to answer, we start by saying the solution is, like distilled water, is hypotonic to the cell sap of plant cells. You must mention the word cell sap. Don't just say the distilled water is hypotonic to the cell. That's not clear enough, right? You must mention cell sap. Now, next, water diffuses into the cell by osmosis, and then explain the effects. So the water will enter the vacuole. So the vacuole expands, and the cytoplasm is also filled with the water. Okay, so when the vacuole expands, it will press on the cell wall and cause the turgor pressure to be produced. This makes the cell turgid. So what are the benefits to the plant when the cells are turgid? It will provide support. It will help the plant to stand upright vertically and the leaves to be extended horizontally to maximize photosynthesis when it's exposed, the leaves are exposed to the sun. And also the gut cells in the leaves will become turgid and they will open the stoma for photosynthesis. So there are benefits to the plant when the cells are turgid. Let's now discuss what happens when a plant cell is placed in an isotonic solution, such as 5% sucrose solution or 0.5 molar sucrose solution. Okay, so first you must mention that the 0.5 m sucrose solution or the 5% sucrose solution is isotonic compared to the cell sap of the plant cell. Always mention compared to the cell sap. Cell sap is a keyword, right? Compared to the cell sap. Don't just say compared to the cell. Right. So water will diffuse in and out of the cell at equal rates. Thus, there is no net movement of water through the plasma membrane, and the cell does not gain nor loses water. Thus, the cell retains its normal size, and we say that the cell is flaccid. It is not turgid. Turgid means that there is a net gain of water, and this causes the turgor pressure to be produced so that the cell is firmer. In this case, the cell is not as firm as the cell in the hypotonic solution. Okay, so for isotonic solution, the cell is flaccid. It doesn't give that much support as a turgid cell. Let's go on to the third solution for plant cell. Let's say you ask this question. Explain what happens when a plant cell is placed in 20% sucrose solution. Now 20% sucrose solution is definitely more concentrated than 5% sucrose solution, which is the isotonic solution to the cell sap of the plant cell. Thus, we can start our answer by saying 20% sucrose solution is hypertonic compared to the cell sap of plant cells. Thus, water diffuses out of the cell by osmosis. Now, thirdly, you mentioned what happens to the cell. Okay, so the cytoplasm and vacuole will lose water. Thus, they will shrink. And the plasma membrane will pull away from the cell wall. So all these lines here are the plasma membrane. Plasma membrane is no longer next to the cell wall. It's been pulled away as the cytoplasm shrinks. However, the cell wall still retains its shape because it's a rigid cell wall. Now, this condition is called plasmolysis from the word cytoplasm. Okay, lysis means to break down or destroy, or in this case, it causes it to change for the worse. Right? So, plasmolysis is the shrinking of the cytoplasm when the cell is placed in a hypertonic solution. This word is very important, and you must know how to draw this picture of a plasmolysed cell. A common question and asked in the exams. However, if the plasmolysed plant cell is placed back into distilled water, deplasmolysis can occur. D means to take away the plasmolysed condition, right? So deplasmolysis occurs and the cells can become turgid again. So the cytoplasm will gain water when it's placed in a hypotonic solution like distilled water, and vacuole expand back again, and the cell can become turgid once more.